History Month, or really any month, by learning about some amazing female scientists. From names you might have heard of, like Rosalind Franklin, to those you're less likely to have heard of, like Hildegard Lamprom. I have a whole page on my blog of links to posts about these scientists and their science. And so there's really no excuse. Go explore. But seriously, there are countless women around the world who have done or are still doing amazing research, and I want to help you explore these stories and this science. In addition to facing obstacles to actually getting into and doing research, women have also had their stories largely been kind of forgotten in history for a number of reasons. Often these women's stories are lost to history because they're kind of overshadowed by the work of a more famous husband or a more famous lab partner. So in the husband's case, we have, for example, Tsuneko Okazaki, who co-discovered the Okazaki fragments. So how DNA, when DNA is getting replicated, how the second strand, the lagging strand, gets replicated in these short little pieces called Okazaki fragments. So until recently, if you Googled her, you would get the little blurb that she was Reiki Okazaki's wife. But they did all this work together, and she actually carried out this work. She continued the work after his tragic early death, when everybody expected her to quit, and she had to get help because she had to raise children. She has this phenomenal story, but you often don't hear about her. Another example is Sylvey Kornberg. She helped with her husband Arthur Kornberg pioneering work in DNA polymerases. She actually helped figure out something that was going on in their reaction that was a stuck point for them, and help them figure it out. Another example of this whole family thing is Elizabeth Roboz Einstein. So she was an amazing scientist, and she discovered the key component of the sheathing around nerves, like the myelin sheathing, this myelin basic protein. But she was also the wife of Albert Einstein's son, and so if you Google her, or if you try to look up information about her or anything, all you find is this information about her famous family. But she even she escaped World War II. She devoted her life to this research, and now this is how she's remembered. Some other scientists have been working in labs of or with colleagues who have big personalities or don't really like to share credit, various stories like that. So for example, Martha Chase of the Hershey Chase experiments that helped show that DNA was the source of genetic information. Hershey Chase didn't even mention her in his Nobel Prize acceptance speech when he was talking about their research. Another example is Flossie Wong Staal. So she was the first to clone HIV. So basically isolate this genetic sequence for HIV, the virus that causes AIDS, and actually stick it into a plasmid. So put it in a form that was studyable, and that she could then stick in cells and show that this virus was actually causing this cellular pathology and all of these different things. And she also discovered lots of stuff about HIV, but she was kind of overshadowed by Robert Gallo, whose lab she was working with him, and he was embroiled in this whole big patent dispute, and she didn't want a part of that, so she left, and she continued to do this amazing work. And so I want to help bring these stories to light and their science to light. So often this situation is even more amplified for women of color, and they're often remembered in terms of being the first to do this, the first to do that. And it's important that we recognize this first, because it shows us how long it's taken, but it's also important that we don't just remember them as the first. For example, Alice Ball developed the first truly effective treatment for leprosy, Hansen's disease. She was the first black chemist, researcher, and teacher, and the first woman to earn a master's degree from the University of Hawaii. But actually she has this tragic story because she died early, and then this colleague tried to steal credit for her discovery, and then this other colleague was like, no, don't do that. He found out years later, and then University of Hawaii had to step back, and now there's a tree named after her and stuff, but she also deserves way more than having this tree. on campus with like a plaque and stuff. So I want you to know about her story and the science that she did, because the science that these women did is truly incredible. And so I want to make it easier to find more about these scientists and their science. So in some of these articles, I have a lot more detailed information about the science. Some of them are more just descriptions of the scientists, a little bit about their work. I used to be the social media chair for the Cold Spring Harbor Laboratory WISE group, so Women in Science and Engineering. So I have like hundreds of posts about female scientists. A lot of them have less detail. I did these like weekly posts for their social media and stuff. And so all of these posts are on my blog, and I want to take you through just like a quick whirlwind tour of some of the female scientists that you can discover, and hopefully it'll give you a little inspiration to go dive deeper into their stories. Often you might be using the results and equations from the experiments done by women scientists without even actually knowing it. So Maud Menton, Michaelis-Menten equation of enzyme kinetics. Yeah, that's her. She's the Menton, Maud Menton. She was this amazing scientist who did these experiments that helped determine this, like how enzymes work to speed up reactions. And you can learn about her and her work in the Michaelis-Menten enzyme kinetics on this page. Martha Chase, who I was mentioning earlier. Yeah, so basically she has this tragic story where she did this incredible work helping discover the DNA was a source of genetic information. And then she was just in this very bad work environment, and Hershey Chase didn't even credit her in his Nobel Prize speech. But you can learn all about that key work from her work in those experiments. So Martha Chase was actually like a research technician in the lab, which is some say that's part of the reason why she doesn't get the credit that she deserves. I should say that not in her case, but in the case of many of the women that whose stories I came across, they were denied promotion or positions of like professorship or faculty positions. And so they were left to having these like lower positions where not only would they make less money, but then they actually were getting less credit than they deserved. And so again, not in the case of Martha Chase, but in the case of many of the women that I came across, they faced these kinds of barriers to advancement that also helped kind of hide their stories in history because they weren't reaching these higher positions. And so their work was not as like valued, even though it was as valuable. Rosalind Franklin, she was another scientist who kind of had her work be not credited enough, let's just say. For example, Tsuneko Okazaki. So I was telling you about her and her work with discovering the Okazaki fragments with her husband. Well, at the time of her husband's death, female scientists in Japan weren't even recognized as like full-fledged researchers. So she had been working in like Rigi's lab. And so when he died, people expected her to like give up research and go back to a more traditional role as a stay-at-home mother. But she even participated in this like citizens campaign advocating for childcare, and she received support from neighbors as well as from other researchers, and she helped pay the way for female graduate student scientists. So she did this amazing work paying it forward, and it's really sad that she isn't given as much, she's not remembered as well as she should be. So she was the PI, so like the head of the lab. Her graduate student, Raymond Gosling, took that fuzzy X picture. And then she was like helping with the interpretation and all of that stuff, and she helped teach him. And so she didn't physically take that photo 51, but she was the the lead author or whatever on this paper, describing their findings that were overshadowed by the work that Watson and Crick did based on that image. Another overshadowed woman is Flossie Wong Staal. So I actually got to see her talk at Cold Spring Harbor Laboratory. She tragically died in 2020, but she was at the laboratory for an AIDS conference, HIV/AIDS conference. She did this incredible research where she isolated, she was the first person to like clone the HIV, so isolate its genetic sequence and stick it onto like a manipulatable, workable form that she could study it. And she demonstrated that one of her clones was biologically active. It was actually her postdoc in her lab that she was mentoring, this Amanda Fisher, who she credits for that. She's sure to give credit where credit's due. She also showed that HIV's genetic instructions are highly variable. So helping like foreshadow like the idea that it's not going to be easy to make a vaccine against this, unfortunately. And she determined the first complete HIV genome sequence and then compared it to other sequences. So she did this incredible work, but as she says in this like oral history she had this like desire to get out from gallows shadow. I was at a stage in my career where I felt that much as I admire Bob as a leader and as a scientist, his visibility was really overshadowing me. I think we had complementary expertise and people recognized that I was doing molecular biology and he was not, but I still think the association sometimes worked against me. And so this is just when she moved to University of California, San Diego, UC San Diego, where she was chosen to lead UC San Diego Center for AIDS Research, and she was elected to the National Academy's Institute of Medicine. She did a lot of really important work, and she's just this amazing scientist that more people should know about. So on the track of HIV research, we have Daria Hazuda, who developed, she did this pioneering work that helped open up a whole class of antivirals for treating HIV. So these like HIV integrase inhibitors, these integrate, more specifically, these like integrase strand transfer inhibitors, or these like INSTIs. And so people had told her that, so this INSTI is basically, it's kind of it's allowing the HIV to get its genetic information into our genetic information so that it can kind of just like get copied with our DNA and hang out. And people thought that the enzyme that did this was undruggable. And so they thought she was crazy for following this, but she was able to lead the development of the first inhibitor for it, of this class, this like raltegravir, Acentris, which was FDA approved in 2007 and is now a staple of combination HIV therapies. So it's even on the WHO's list of essential medicines. And so you can learn more about that here. Still in the HIV story, we have Nemanja Bumpus. And so I actually got to meet her too. So she is this amazing pharmacologist, and she is actually director of the Department of Pharmacology and Molecular Scientists at Johns Hopkins. And so she, at the time I wrote this, at least, I don't know about currently, she was the only Black woman chairing a pharmacology department at any medical school in the entire US, which is like crazy. That's just not right. But anyway, she does this amazing research studying how like the body metabolizes or like processes drugs, including drugs used to treat HIV, and showing that there can be genetic differences that are more predominant in certain groups of people, and that we really need to take this into account and really incorporate this genetic diversity into like research, clinical trials and that sort of thing, and really study more diverse populations in order to see if how drugs might affect different people differently, and make sure that we're treating everybody appropriately. So Theodore Haciano, so you might have heard of her more recently for her work studying antibodies against COVID-19, but she actually did some pioneering work on HIV/AIDS. She did what she's said to have like broken the species barrier and became the first person to develop a non-human or chimp AIDS model. So before, people have been studying like monkey-specific types of viruses, like simian virus, simian immunodeficiency virus, which is similar to HIV, but it has differences. And so if you want to study the human virus, you need to be able to study something like HIV. And so she found this barely modified version of HIV that she was able to use to infect this type of monkeys. And so she was able to get this to work and have developed this model, this non-human primate model of HIV/AIDS, and therefore study it and study potential treatments and that sort of thing. Well, on the topic of viruses, so the mRNA vaccines against SARS-CoV-2, the virus that causes COVID-19, those were made possible by the work of Katarina Kuriko. And so she actually developed this whole kind of made mRNA vaccines possible. People didn't think it was possible. And they didn't even they stopped funding her. So she believed in the synthetic mRNA's potential, but she was actually demoted by University of Pennsylvania because she couldn't attract funding. And they said, like, change your topic or we'll demote you. And so she didn't want to change her topic. She really believed in this mRNA technology. And so she took the demotion and then published her groundbreaking paper, but then it got little traction. UPenn even refused to undemote her, and so she and let her have a tenure track position. So she left and joined BioNTech, a senior VP, and now she is basically responsible for helping save a lot of lives. And you can learn more about her and her work on this post. Speaking of saving lives, I don't have as much about her science here, but Ursula Franklin, she's just like this incredible woman. She was a Holocaust survivor, and she was shipped off to a forced labor camp, and her parents were sent to concentration camps. So thankfully they all survived and they were reunited after the war. And she went on to study radioactivity and investigate like levels of radioactivity in children's teeth, leading to bans on nuclear weapons testing. She was a pacifist and a feminist, and she helped successfully sue the University of Toronto as part of this advocacy group, Voice of Women, and which is now the Canadian Voice of Women for Peace. They sued the University of Toronto for paying women faculty less than their male colleagues. And so she was this amazing woman. Here's Elizabeth Rose Einstein, who I was telling you about. So she isolated and purified this like myelin basic protein, which helps keep that sheath together. And she like investigated its potential role in neurodegenerative diseases, such as MS. And so she has this, she also had, she faced, in addition to sexism, she also faced anti-Semitism, and she has, you can learn more about her story here. One of my favorite female scientists is Jane Richardson, who I have also had the pleasure of meeting. She is a crystallographer, and she developed that ribbon diagram notation for displaying protein structures as like, so alpha helices as these like ribbons, and then these like beta strands as these flat arrows, and then the beta pleated sheets where you have these beta strands together. And you can see more about how she developed her process. And she also developed this software, Mold Probity, as well as some other software that helps structural biologists, so the people like figuring out the protein structures, helps them analyze their work and check that it's like fundamentally sound. So she has this very, she sees the beauty in these molecules, but also wants to make sure that they're kind of like anatomically correct at the atomic level, and that all the geometry is matching, and so you don't have clashes and that sort of thing. And she is like almost entirely self-taught in all of this stuff. She is just really, really amazing. And so she actually helped work with her husband, David Richardson, to solve some of the first protein structures. She's also kind of one of my heroes because she contributes to Wikipedia, and she makes her like figures available on Wikimedia Commons, so people could use. She even got the RCSB PDB to make their molecule of the month graphics similarly accessible. And so, yeah, she's just
really, really cool. Eileen Southgate, oh my gosh, like I cannot imagine what this must have been like. She helped map all of the neurons in C. elegans. So in these like in these worms, these like nematodes, she traced by hand like all of these different slices of worms and like labeled and tracked which nerve, like in one picture and then the next picture, which like nerves and cells were connecting. So they found a total of 8,000 synapses, so where the nerve cell meets another cell, and included about 2,000 neuromuscular junctions, so nerve meets muscle, 5,000 chemical synapses, and 600 gap junctions, so like electrical connections. So this was like she was working as a technician or assistant in the lab with John White and Nicole Thompson, and in the lab of Sidney Brenner, who were doing all of this. And so she did a lot of that rote tracing work and all of those careful figure makings. It's really just like this incredible story. She also worked for a lot of other, did a lot of other crucial research in different labs. Hildegard Lamprom, she helped develop like the first in vitro translation system, so basically being able to make proteins outside of cells. And she was able to study how protein gets made and show that proteins, that mRNA was telling these ribosomes what proteins to make, and that the ribosomes were kind of like associating as these polysomes, so these like multiple ribosomes working together, and that it wasn't just like an aggregate, that was actually the functional stuff. So she has a really cool work here too. Sylvey Kornberg, so basically she did this crucial work with her. So her husband, she was the wife of Nobel laureate Arthur Kornberg and the mother to Nobel laureate Roger Kornberg. So it's hard to find information about her. And you often find information about her family. But she actually helped with some of the research and did important research of her own, and helping like with their research and then also finding other things. And so in this picture, so this picture, like basically readers on social media helped identify her in this picture because she was shown as like labeled as a pharmacy technician, and then researchers from social media helped identify her. So that's just another fact about her. So Dorothy Crawford Hodgkin was a pioneering X-ray crystallographer who made it possible for a lot of, basically all of the protein crystallography work that is done today. So she saw the structure of penicillin as well as vitamin B. She did this really crucial work helping make possible all of the work that is done today. And she is she is like the first of a long line of influential and powerful female crystallographers. So if you ever wonder why like folate, sometimes in the form of folic acid, is added to bread and stuff, that's because it is a cofactor or basically a helper molecule for various enzymes, so reaction speeder upper helpers. And so this is sometimes called like Will's factor. She was able to find this dietary connection between a disease of pregnant women in India, and show that if you gave them this supplement that contained what was later found to be folic acid, this could both prevent and help reverse the disorder. So more about her here. Here's that post on Alice Ball and her work on developing this like ball method to help. Basically, the shawl mover oil was used to treat leprosy, but oils are like all goopy and not good for injecting into people, and people were getting sick if you were just giving it to them directly. And she found this way to basically break it up into these parts and then modify them, and then this could be taken by people with leprosy and be able to help treat their disease. Patricia Bath, she was the first Black female doctor to patent a medical device in the US. She developed this like minimally invasive laser-based method of removing cataracts. It's called this like laser phacoprobe. And she also established this discipline of like community ophthalmology, which views basic eye care as a human right and works to provide it to everyone. And so she took this like holistic view to trying to figure out why there was this disparity in Black people being more likely to suffer from blindness and to develop glaucoma. And so people were looking to try to find genetic roots, and she took this more holistic view. She had grown up in poverty, and she knew that people could, that this affects access to care. And so she helped to make it so that people could have more easily get care for their eye problems, establishing this idea of kind of this community ophthalmology, viewing it, this eye care as a human right. So there are a couple of women I have posts featuring on their work on cystic fibrosis. So Johanna Ramans and Batsheva Karem. So Johanna Ramans helped discover the gene, the CFDR gene, which when mutated is responsible for causing cystic fibrosis. And so did Batsheva Karem, who was an Israeli geneticist who helped discover this gene and the most in finding the globally most common mutation in it, and helping try to like develop targeted therapies for it. You might have heard of like Nelson diversity surveys. Those are thanks to Donna Nelson, who is a organic chemist, and she also is known for drawing evidence-backed attention to the lack of gender and ethnic diversity among faculty in top science departments. So this is where these like Nelson diversity surveys come in. So basically people had known that this underrepresentation was this huge problem, but they didn't have the proof to point to, and so she helped develop this way to get that actual proof. She's also a native scientist, and she is active with SACNAS, so the Society for the Advancement of Chicanos, Hispanics, and Native Americans in Science, which is a really great institution that I encourage you to check out and donate to. So Marion Sewer was a pharmacologist and professor at UCSD Skaggs School of Pharmacy and Pharmaceutical Sciences, and she researched steroid hormone biogenesis. She had this tragic early death and from a pulmonary embolism. But before her even in like her short life, she was able to carry out this critical research on how these steroid hormones are made. And she was super committed to increasing diversity in science. So she was a tireless advocate for underrepresented minorities. She helped co-found and lead grant writing workshops in a membership program to help advance the careers of underrepresented minority scientists. And she has a lot of really amazing work that she has done. And there's now like an ASBMB scholarship awarded in her memory. CRISPR, so that gene editing tool, for that we have to thank Jennifer Doudna and Emmanuel Charpentier. And so you can learn more about CRISPR and Jennifer Doudna. I didn't have enough information about Emmanuel Charpentier. I need to learn more about her, but I'm sure she's really awesome too. But I actually got to meet Jennifer Doudna, and so that was really cool. Another really cool scientist is Lana Banga. So she helped discover actomyosin, which is the like actin-myosin combo that makes your muscles contract. And she also did this like super incredible work helping save the lab during World War II. And so she was left to protect the lab and all of its valuable research equipment. And she posted notes on the door in like all these different languages saying that it was researching infectious things. And providing sample drop-off hours for infectious materials, and this scared away anyone who wanted to take their equipment. And their Institute for Medicinal Chemistry maintained intact. And she did this incredible work both with actomyosin and also with elastase, so this kind of enzyme that was causing this like degradation of the elastin fibers, like holding together the walls of veins. So she had this like incredible number of papers, And now she has a Wikipedia article. And so basically, she was one of the first female scientists that I made a Wikipedia article for. The very, very first was Virginia Minnick, who discovered hemoglobin E, which is this abnormal form of hemoglobin that can cause blood disorders. And she worked out this like glutathione synthesis pathway. And so I'm bringing this up not to like say like, see, look how great I am, I made these pages, but just because this was how I learned about a lot of these women, was making these pages, and I made them so that other people could learn more about them too. And the reason why I'm saying this is because I want you to help. You can write, you can make Wikipedia articles. Anybody can edit Wikipedia articles. I have resources on my blog to help you. And it's a really, really great way both to learn and to help others learn. And I have to thank for this Jess Wade for helping inspire me. She's written an astronomical number of articles on female scientists and really inspired me to get started. So speaking of getting started, in addition to getting started with Wikipedia editing, you can now get started exploring some of these amazing female scientists. And I hope that you are able to learn and be inspired and search out and find even more.